Our last speaker of the evening before we go into the panel component where we'll invite any of your questions and hear what the speakers have a discussion amongst themselves is Dr. Michael Kobor, standing right here. He's a professor at UBC in the Department of Medical Genetics and the Human Early Learning Partnership. He's a Tier 1 Canada Research Chair in Social Epigenetics, Sunny Hill BC Leadership Chair in Child Development, and the Theme Lead for Healthy Starts, a BC Children's Hospital Research Institute. Currently, Dr. Kobor's research is investigating epigenetic variation in humans with a focus on the effects of social environment on lifelong health and aging. These effects are continuously explored through ongoing collaborations with psychologists, population health scientists, and experts in fetal alcohol syndrome, asthma, and other health conditions. Dr. Kobor.
Thank you. So it's a real pleasure to be here, and hopefully I'll manage to bring together some of the exciting themes that we heard from the first two speakers, and perhaps provide a bit of a molecular substrate for some of the things we heard out of the telomerase. But I want to just start with a small story. And the reason I'm here, I think, is that over the August long weekend, we did a lovely hike in the subalpine in Manning, and some fellow comes along with a heavy drum on his neck. So, this started chatting with us. Taylor here, so thanks for having me. It's a real pleasure. So what I'd like to do today is really talk with you about epigenetics and share some of the sort of foundational data why epigenetics has emerged as a key interface between the genome and the environment. Then show some of our own empirical data that really speak to epigenetics really being a biological vestige of early life experiences, and sort of going quite a broad spectrum, ranging from poverty to family environments and all the way to child abuse. And if we have time, I'll touch upon a sort of neurodevelopmental disorder that where we can also see some epigenetic associations that being fetal alcohol spectrum disorder. But I think what I'd like for you to take away from this particular lecture tonight is that epigenetics is really emerging as one of the key interfaces between nature and nurture. And there are really many different factors, perhaps consistent with how messy human life can be, that are shaping the human epigenome. So, what is epigenetics? Well, I grew up in Germany, and Germans are supposed to love definitions. I actually hate definitions, which is why I grew up in Canada. So for those of you who like definitions, this is perhaps the most informed definition of epigenetics. What we're really trying to focus on is this particular aspect on how life experiences actually affect the way our genes are expressed. And the paradigm that we're really operating under is this idea that we have experiences and there are particular biological pathways that are affected, and that then drills all the way down into the level of the nucleus of the cell to affect the expression of genes. Of course, it's not a one-way road, but it can also go the other way back again. Now, to understand epigenetics, what I want you to just focus very briefly on is this is a piece of DNA. Of course, the DNA actually doesn't occur like this in our body. It's actually wrapped in many different ways and modified in many different ways. And all of those modifications are actually what underpins the epigenetics. And so there are many different processes and layers that really shape what we call the epigenome. And they're all linked to this nuclear DNA structure called chromatin. And they're all listed here. Really, all of them have a terrifically interesting story to tell, but we don't have time. There are actually textbooks just filled with all of those. So what we're going to focus on tonight is really one of them, and that is the methylation of the DNA. That's a particular small chemical modification by which that's attached to the DNA in a particular context of often, but not exclusively, in these CpG dinucleotides. And it really has become the workhorse for human population epigenetics because it is very accessible. It can be measured quantitatively and at a fairly reasonable cost. So I can get your epigenome for $300. If you all give me a sample by the end of the day, please report to my research technician outside. And collectively, then the insights of all of these studies have really led to the finding of the emerging notion that there are many factors that shape the DNA methylome, including, of course, our own development, but also environmental factors. And so this is probably the most molecular slide for you tonight. So I just want you to take a step back and really think of this DNA methylation and the gene expression in a more simple way. So for me, essentially the way I like to think about this is that each gene is a light bulb. And as you know, a light bulb can be either totally on or totally off, or if you live in a fancy house, it can be anywhere in between because you have a dimmer. Now the dimmer is actually, in this case, the DNA methylation. And particularly when this DNA methylation happens to occur in the regulatory region of the gene that then regulates this expression. So how can we think about this? Well, pick your favorite gene. Now this favorite gene is very much on because we have no methylation, and this is a gene regulatory region. It's unmethylated. Now as we begin to methylate, you see we are pushing this dimmer down. We're starting to methylate, get more methylation and even more methylation. As we methylate this gene regulatory region, we're pushing that dimmer down, we're turning the light off. And so that's really all you need to understand and how DNA methylation works for the purpose of this talk. Now one of the really interesting opportunities that we have in epigenetics is that actually our epigenome, in contrast to our genome, which we inherit from our parents at birth or at conception, is not stable. And in fact, our epigenome changes from the beginning of the human development all the way to old age. And one of the key factors that distinguishes the epigenome from the genome is that it's very different depending on which tissue and which cell you look at. So here are two of my favorite cells. Anybody has a wild guess what they are? I hear neuron. These are very good. Qualify as a neurobiologist. Anybody knows what this ball-like structure might be? Did I hear blood cell, lymphocyte? No? Okay, so obviously I think we all agree that these cells look very different. They do very different things, and yet they have the very same genome if they're from the same person. And so the way we can imagine this is that they have essentially different light bulbs patterns. And I apologize, my graphic team I couldn't get them to draw 25 light bulb instead of the 25,000 that the human genome has genes, but bear with me. But so there are different light bulbs in these different cell types. And when we talk about the environment affecting and associating with different epigenetic patterns, we're not talking about the environment changing a neuron into a blood cell. Instead, what we're talking about, that it might in subtle ways change a few of those light bulbs. So in some cases, it could be that it does that so in both tissues. In other cases, it could be that it might differ depending on which tissue we look at. So with that being said, I want to show you one really, really exciting example. It's an older example, and still is perhaps to me the best example of what epigenetics really is. With that, we switch to Lisa's favorite rodents here. When you see these five, these are very young mice. When you see them, what would you naively say what's different between them? Color. Right? Does anybody think the color is the same? Good, otherwise we have to send you to the optometrist. Now, when you see sort of different colors, why usually would be a coat color different? Different genes, right. Well, it turns out, and don't ask me how they do it because I'm not a mouse biologist, and we can talk with Lisa. These are actually genetically identical mice. So some mouse person has bred them down in their basement to be exactly genetically identical. They were born to different mums though. And the reason what happened is that the reason they look different is that the mum was actually fed a different diet during pregnancy. And there's one particular gene that gives rise to this coat color called the agouti gene, where the upstream gene regulatory region actually determines, as we said earlier, its activity. And in this particular set of mice, this gene is totally turned on. There is no methylation, the dimmer is all the way up. Now, if you look over to the right side, to the more brown-looking mice, what we see is that we're pushing this dimmer down. We're getting more and more methylation to end up with the light bulb being totally off and this mouse looking like this. Now, there are a number of components of the maternal diet, including things like folate, but also alcohol and many other bisphenol A that can, in subtle ways, regulate the activity of this upstream region and the activity of this gene through DNA methylation. Now, of course, you know, most of us don't really care whether we have a mouse that looks like this or like this, or is anybody really passionate about this? I sort of just want to keep them out of my kitchen. So the question then becomes, what happens with these mice when they grow up? And I'm going to sort of just put it out there. Any wild guesses? They do behave differently.
One of them doesn't grow up.
Okay, what about this? So you see those two mice, genetically identical. The only difference was their mom was fed a different diet during pregnancy. This fellow is obese, develops all kind of age-related health problems. This one is lean and fit. And as Lisa said, they also actually develop different behaviors. So really highlighting the power of epigenetics, because what you have here is you have an environment during pregnancy that literally gets under the skin of these mice to change the activity of one gene, of some number of genes, through different methylation of their gene regulatory regions. And it does so in a very persistent way. It sticks with us, or with these mice, throughout their lifetime to profoundly affect health and behavior across the lifespan. Now this has been, similar effects has been seen in humans. This is some example from the Dutch famine, where several studies have documented that there is a lasting effect depending on whether the women during pregnancy were starved or not. There are similar studies that have documented a signature of prenatal exposure to smoke, whether it's passive or cigarette smoke, active or passive, in the offspring in humans. And we and others have started to see epigenetic signatures of alcohol exposure that I probably won't have time to talk to you about tonight. Now, we heard a very exciting and talk by Adele about the sort of, and Lisa about the quality of maternal care. Seminal work by our Canadian hero Michael Meany and colleagues has actually shown that in rodents and later on in humans, there is an epigenetic association with the quality of the maternal care that a child receives. And of course, the closest we can ultimately come to these mice that I just showed you are identical twins. Anybody knows identical twins in here? Are they very similar, always the same? Or? Depends. No. So, identical twins, like these two cuties here, one happy and the other one sort of screaming away. And a number of interesting studies, initially by Alpatronis in Toronto, but most recently by Jonathan Mill in England, have shown that identical twins often start off with a very similar epigenome, but it gets discordant as they go through life. And then Jonathan in particular has shown in a number of ways that this might be associated with differences in mental health outcomes, including schizophrenia and depression. Now, taking all this then together in my own laboratory, my own team, what we really try to do is to apply this idea of epigenetics to child health and really to understand the interface of nature and nurture. And what we're trying to do is to really focus, of course, on the interface, on the connection between the environment and epigenetics. I'm not going to have time to talk to you about it tonight, but we also were among the first to show that the axis between genetics and epigenetics is actually much more prevalent than initially anticipated. And of course, you've heard of many different gene bioenvironment studies. But collectively, we try to bring this together, the nature, the DNA, and the nurture, the environment, to then understand how can we give our children the best start in life, and how can we ideally prevent diseases. And historically, one of the main areas that we've been focusing on was socioeconomic status, to really try to understand how early life experiences get on under the skin, and also early life parental stress. And this started sort of approximately 10 years ago here in Vancouver, and over the years we've been really standing on the shoulder of giants to do a lot of this work. You can see some of the key names of our partners here. We essentially have the hypothesis, set out this work with the hypothesis that indeed there are differences in these light bulbs and dimmer switches that relate to some of these conditions. And what I didn't tell you and what I didn't tell you, my actual training was in yeast genetics and yeast epigenetics. And so when I sort of started getting involved in particular with Craig and Edith, it's like, oh, this is sort of interesting. But when you actually look at some epidemiological studies, and Adele talked about some of them, it's actually quite prevalent. For example, what a powerful predictor of early health, early life socioeconomic statuses, and that's really an area that, as you know, Greg and Youth have been focusing on. And there are many different conditions that that can be seen, but one of the most best powered studies really showing here that there is a difference in the heart disease in the American physicians, male physicians graduating from Johns Hopkins University. This was before NIH said that you have to do females and males. But with a difference between 2.5 times higher probability of heart disease in physicians, regardless of their current status, that grew up in low socioeconomic status during the first 5 years of life. So there are many of these studies suggesting that it really matters how you grow up, and Adele talked about some of them, of course. But really, I want to just pause here for a second and give credit and really acknowledge and highlight the amazing work and foresight that the late Clyde Hertzman from the Human Early Learning Partnership had in all of this. As many of you know, Clyde was a champion, never tiring champion of children, a wonderful mentor for many of us, and really such a powerful advocate. And even before all of this idea of epigenetics came about, Clyde already published that there was a biological embedding of early experiences that really had profound effects in adulthood. So I just want to give credit to Clyde, and getting to say that he's not with us anymore. And so really, of course, this is also linked to this idea that there is a developmental origins of health and disease that has become more and more prevalent even in the public. Now, we then tested some of these predictions in that the first study we did some years ago with Greg and Edith really took 100 healthy adults from the Vancouver area. And what we did, we just essentially tried to recreate this Johns Hopkins physician study in our own way by having two groups that were separated by low socioeconomic status and high socioeconomic status in the first 5 years of life. So, naively speaking, growing up somewhere here versus here, and then mixed in adulthood so that we would not get confounding data by adult socioeconomic status. And we looked at a number of things. We first looked at the light bulbs. That was at the time the easiest to look at in their white blood cells. And what we indeed found that there were differences between those two groups, and there weren't random differences. Instead, when we sort of looked, some fancy bioinformatics, there were differences that could be linked to particular transcription factors regulating sets of genes that were predictive of having a higher inflammatory response in those people that grew up in low socioeconomic status. And in fact, when we then tested their immune system functionally, we found exactly that the prediction was correct from the gene expression profiles, in that their immune system in all cases, the innate immune system, these toll-like receptor pathways, actually acted more strongly. You could argue perhaps overreacted. And some of you might know that an overactive sort of latent immune system is actually not a good thing, because it has been linked with a number of problems, including heart disease and mental health problems as well. Further, when we looked at cortisol output, we also found that there was a change in cortisol with the folks who grew up in lower SES, having again higher cortisol output throughout the day. Some years later, then we were able to look at the dimmers themselves, because the technology had advanced sufficiently for us to do this. And so what we then reported in another PNS paper 3 years later is that there's actually, as judged by this highly skewed p-value distribution, an association of early life socioeconomic status with the white blood cell DNA methylation pattern, these dimmers in these adults, but not, interestingly, with adult socioeconomic status. So at least in that study, it suggested that when we sort of look at us in the room, there's a signature of how we grew up the first 5 years of life, socioeconomically speaking, but not as much as the socioeconomic status that we have here. Again, consistent with the idea that our experiences get under the skin to stay with us potentially for the rest of our life. Of course, these are sort of provocative studies. These are the first we did that, so we're constantly trying to look at different conditions, but also trying to replicate the same ones. And in this particular case, we turned to Tom Boyce, my lovely colleague who was here at UBC for a number of years, and Nikki Bush, with the Peers and Wellness study, which is a children's study in Berkeley, California. And in this study, it's a longitudinal cohort where we have a more nuanced measure of socioeconomic status than the one we had in the initial study with Greg and Edith. And this is actually coming out tomorrow, I believe, this paper. And Kath, my knowledge translation specialist, is going to do a Twitter feed on it, I think with the figures, so stay tuned on our Twitter. But what we found in this paper, again, is highly skewed p-value distributions and some statistically significant correlations between a number of SES-related factors, including income-dependent parental education, but also family adversity. Interestingly, those seem to be largely distinct epigenetic signals, but there are also some overlaps. So we're really happy about this work because it's for the first time we're trying to really tease apart these very complex situations. Now, we also try to understand whether there are other environments beyond this powerful socioeconomic status and ones that might be associated with the human epigenome. And we already heard from Adele that we should love our children. And so we actually were lucky enough to sort of have a situation where we could more or less replicate a famous rat study with Michael Meany. And this is because my wonderful colleague Ron Barr, who just retired, and along with Tom Boyce, actually had a cohort of children that they had measured in real time for a week the contact time between the caregiver and the child. And you can see that here. It's actually quite remarkable. If you look at this distribution, it goes from something like 300 minutes almost to like 24 hours for some people. And so what we did, we got DNA from these kids over here, from these kids over there, and we simply asked, is there an association with their epigenome that depends on the contact time? And long and behold, as my wonderful postdoc Sarah Moore published last year in Development in Psychopathology, we identified a number of regions where there was a difference in DNA methylation that could be related to contact time. We also, she also identified a new measure of developmental age that was associated with contact time, in particular in kids that were very fussy. And so you can read more about this. But of course, you know, similar to what Adele said, the Vancouver Sun then, of course, creates this nice headline, "Cuddling's a baby or not affects infants' gene expressions." It's all good. Cuddle. Now, aside from these sort of more feel-good things, we also have been wading into more perhaps challenging territory, that of child abuse. And we started a collaboration with folks at Harvard on their gut study, growing up. It's an offspring of the nurses study. And when we started this, it was very challenging. It was the first time we looked at sperm cells. And this is a rather small study. It was published just a few weeks ago. You might have seen it. Again, it sort of got lots of headlines in CBC. The National Post put it together with the Kavanaugh story. So it's not... It was kind of interesting. We can talk about it over refreshments. But what my very talented student, Nicole Gladish, found is that when she does some sort of principal component analysis, you can find here that in this fourth component, which explains roughly 7% of the variance of this methylation in the sperm, that there was association with abuse level, with whether it's continuous or not, and with the CTQ, which is a measurement of adversity. And she also was able to identify particular gene regions using two different methods where you can go see very strong effect sizes, probably the strongest I've seen in the sort of 100 studies that we have done in the sperm between these men that have been heavily abused and the ones that have not. And again, this can be seen and we demonstrate this by two different methods. We sort of, so to me that's a very interesting story that you know, I look forward to talking more about later on and what this all means and how we can use it to perhaps help people, or how it can be potentially be abused. And so, you know, as I said, we constantly on the lookout for interesting studies and trying to sort of use natural experiments to really try to expand our view of what's called the epigenome. So I just have two here, where we have shown in the first one together with Megan Glenner in Minnesota that there is a signature in children that were adopted from East European orphanages in the first 2 years that I lived for the first 2 years of life to North America, Minnesota in particular. Together with Tom McDade and Chris Kusawa, we have been doing some very interesting work in the developing world in the Philippines, where we have documented both social and physical environments early in development, also predict DNA methylation in adulthood. And sort of putting this all together, then together with Tom, we've built on this initial work, this initial seminal paper by Glide, to, in a recent review, to really expand the view that the epigenome is really at the synapse, if you wish, between the gene and the environment interplay. And of course, you know, from the elegant title, that's Tom's title, not mine. I'm going to switch over to fetal alcohol work to just sort of summarize what I've shown you today. So actually, this is a slide I just got this afternoon. I had a different one, but my students and trainees actually are very creative and they put their own pictures in here. And coincidentally, those are actually the folks who did the work. So what we show here is that there is obviously that the epigenome lies at a different interface between experiences, and this is Nicole who did the work on the sperm DNA methylation and phenotypes, and this is Rachel who did all the work on the epigenetic, the paper coming out tomorrow, and so if kids that are very, exuberant, or the kids who are more retrieved, this is Sarah who did all the work on the attachment. And of course we need to look at this in the context of development. We need to consider tissue specificity, but we also want to consider our genome. And we've also started to sort of think about this in a more sort of holistic terms, and sort of how can we put this work into better use, and started to think about how this could affect potentially child health policy. So we can again discuss this later. But I just want to leave you with, is it nature versus nurture, or both?